Good afternoon and a virtual welcome to my talk today. I would like to share our recent study with you about the use of target-directed dynamic combinatorial chemistry for hit optimization of anti-infective target DXS. A recent analysis of published clinical candidates shed light on which lead generation strategies are most frequently employed in identifying the drug candidates. And the most frequently used lead generation strategy was based on a starting points derived from previously known compounds, followed by a random high throughput screening, and the remaining approaches included structure-based drug design, focus screening, fragment-based lead generation, and in some cases DNA encoded library screening. It's worth to note here that the quality of the starting point can influence the drug discovery process, and a high quality hit can shorten this drug discovery path to a great extent. Target-directed synthesis approach can generate these kind of high quality hits as biological target is involved to assemble its selective ligands. These robust strategies are recently gaining the popularity in early drug discovery projects, and they can be divided in two major approaches. So the first one is thermodynamically controlled target-directed DCC, which involves reversible reaction and where compound libraries can be generated from appropriate building blocks. The addition of the external stimuli like target protein can alter this equilibrium and change of composition of this library occurs. In DCC, in target-directed DCC, the target protein can select its own binder and favors their formation over the non-binders, thus resulting their amplification in the library pool. The second approach is kinetically controlled reactions involve irreversible bond formation, and it's known as kinetic target guided synthesis. Here, target catalyze the irreversible synthesis of its own ligands from the pool of building blocks. This also results in the amplification of the binders in the library pool. So here I would like to focus on target-directed DCC, and there are two main approaches to run the target-directed DCC. The first one is the comparative approach, where a blank reaction without target is run in concurrent with the protein templated reaction. So after a given time in protein templated reaction where the reaction equilibrium is reached, the reaction equilibrium is frozen and protein should be denaturated to release the bound ligands. This action is also needed to avoid the library to re-equilibrate and avoid the protein to further amplify the binders. Then the comparative analysis of the blank and the templated library will provide the information about the amplified hits in this comparative approach. Another approach of DCC is non-comparative, in which the hits can be analyzed in complex with the target or after being released from the target. There are different techniques that can be used to analyze these DCLs, which also includes quite commonly used LCMS or size exclusion LCMS and NMR spectroscopy. And in some cases for the complex studies, X-ray crystallography was also used. Comparative to the activity-directed synthesis approach presented by Professor Nelson, only a limited number of reversible reactions have been used so far in the target-directed DCC experiments, out of which imine formation, acylhydrazone, and hydrazone formation are widely used, followed by thiol disulfide exchange, boronate transesterification, Michael type of addition to enones, and in some cases, hemithioacetal formation as well. So even though the reaction scope is limited, the target-directed DCC approach is used to identify hits of several important therapeutic targets. Here is the overview of recent biological target used in DCC. This biological target includes anti-infectives, neurodegenerative, cancer, inflammatory, parasitic, and DNA, as well as protein-protein interaction targets. Nonetheless, to the best of our knowledge, the use of target-directed DCC has been limited to the initial hit identification process. We decided to explore the possibility of using target-directed DCC for hit optimization. In the target-directed DCC, target selects and amplifies its binder from the library. Thus, it can be used for the further optimization of the initial hits in subsequently tailored libraries inspired by the initial hits. For example, if we use the building blocks from the initial hits along with the new building blocks, there are chances of finding better hits in the next round of DCC. We decided to test our hypothesis on anti-infective target DXS. Why DXS as a target? Actually, DXS catalyzed the first rate-limiting step in the MEP pathway to form the DXP, which is a precursor to vitamin B1 and B6, as well as isoprenoid precursors. So inhibiting this enzyme, which lies on the branch, can inhibit several biosynthetic pathways which are essential for the bacterial survival. Moreover, the MEP pathway is one of the two known pathways for the biosynthesis of terpenoid building blocks, and it is absent in humans. So developing inhibitors of enzymes in this pathway could give us a chance to develop a potential selective drug candidate. For our study, we used acylhydrazone formation reaction for the DCC experiments. The selection of the first dynamic combinatorial library was based on some structural similarity of the building blocks with the cofactor thiamine diphosphate. So it's important to note here that structural information of cofactor or enzyme or inhibitors is not at all necessary for running the DCC experiments, and even a random selection of building blocks could also be possible. Here we have selected 3 aldehydes containing pyrimidine and pyridine moiety, and 7 different hydrazide, mostly featuring various heterocyclic cores. So in total, we would get 21 acylhydrazone products from this library. And we decided to dock these acylhydrazone products in the active site of DXS using LeadIt and CSAR software from Biosol IT. The docking study reveals that most of these acylhydrazones are accommodated in the active site of the enzyme DXS. So we performed the first round of DCC experiment by reacting 3 aldehydes with the 7 hydrazides in presence and absence of the protein. So comparison of these DCC experiments in presence and absence of the protein showed us that there are 5 acylhydrazones which were amplified in these runs, and they were amplified to the different extent. So for example, if you look at the compound 1, which was amplified the most, and compound 5th, which was amplified the least. So in other words, that could mean that the compound 1 binds better as compared to compound 5 to the enzyme DXS. So we synthesize all the 5 hits from the DCC1 and evaluated their binding affinities for DXS using surface plasmon resonance. The 5 hits showed moderate binding affinity ranging from 55 to 270 micromolar. Interestingly, the trend of binding affinities of these hits mostly correlated to their amplification in the DCC runs. Furthermore, we also evaluated the enzymatic activity of these hits against the DXS, which showed a similar trend that the most amplified hit was the most active one, showing 51 micromolar IC50. Unfortunately, when we tested these compounds for their antibacterial activity, they showed maximum of 24% inhibition of E. coli at 50 micromolar concentration. Nonetheless, using target-directed DCC, we were able to find a new class of inhibitors of anti-infective target DXS, which provided us a good starting point for further optimization of these hits. For this purpose, we, instead of using the traditional metchem, we use target-directed DCC and let the target protein select its best binder. So we assume that using the common structural motif from the first round of DCC hits as a building block in the second round would give us improved binder or inhibitor in the second round of DCC. So from the initial hits, tetrahydrobenzothiophene and indole are emerged as a true privileged structure, which are present in first 3 hits and 2 hits respectively. So here we wanted to investigate the effect of inverting the position of these 2 groups in acylhydrazone. To do so, we included their corresponding tetrahydrobenzothiophene and indolaldehydes instead of hydrazides. Along with these 2 aldehydes, we included bromopyridine aldehyde and 4 new hydrazides along with the 3 old ones. So when we analyze the second round of DCC experiment and compare the blank reaction with the protein templated one, we found out the amplification of 5 new hits. These hits were amplified to the different extent, but comparatively to the hits from the round 1, the amplification was way higher. So we synthesized all 5 new hits and evaluated their binding affinities for the DXS by SPR. The KD values of these 5 hits were in the range of 7 to 150 micromolar, which is a significant increase compared to the first 5 hits from the first round of DCC. However, when we tested them against the DRDXS for their enzymatic activity, there was no significant improvement. But when we tested for them for the antibacterial activity, we saw a significant improvement in the antibacterial activity, and compound 9 was the most active one, showing 19 micromolar MIC value. This improvement in the binding affinity and the antibacterial activity confirm our hypothesis to a certain extent that DCC can also be used for optimization of the inhibitors. To further optimize the hits, we performed another 2 tailored DCC runs using the common structural motif from the potent hits from DCL2. So we selected 2,4-dichlorophenyl and thiophene from these hits and included them as a corresponding hydrazides in the next DCC run. We also sought to unravel the influence of flexible linkers on the activity of the hits and thus include the corresponding hydrazide with the flexible linker. Another common structural motif which was present in the active hits from the first and the second DCC run was also included, and that was indole. So here we use indole 6-carbaldehyde instead of indole 3-carbaldehyde to understand the effect of substitution pattern of this ligand on the binding to the protein, and if it also influenced the activity of the potential acylhydrazones. So in the third round of DCC, along with this 2-hydrazide, we included 22 commercially available aldehydes. For operational simplicity, we divided it into two groups of 11 and reacted them separately to these two aldehydes. Another DCC which was focused on 6-carbaldehyde indole, where we also included thiophene and pyrrolaldehyde. And to know the effect of inverting these groups in the acylhydrazone, we included them as aldehyde instead of their hydrazide counterparts. Here also we aim at studying to some extent the SAR using the DCC approach by including a range of heterocyclic building blocks to cover a wide chemical space with a decent structural diversity. On the contrary to the traditional metchem approach, where you would possibly synthesize all the 68 acylhydrazone compounds and test them for their biological activity, we decided to let the protein select its best binders and synthesize only those which are amplified in the DCC runs. The analysis of these target-directed DCLs revealed the amplification of total 29 hits. The carefully inspecting the hits from these three DCLs revealed that 2,4-dichlorophenyl moiety and 5-member heterocycles are favored. As hypothesized, 6-carbaldehyde indole instead of the 3-carbaldehyde one was well tolerated, given that 5 hits containing these structural motifs were amplified in these DCLs. Moreover, several hits containing different substituted phenyl rings were also amplified. We decided to synthesize a total of 11 out of 29 hits based on their representative class, which includes 5-membered and bicyclic heterocycles, functionalized phenyl rings, and chiral compounds. We also took into account their amplification in different DCLs, and we evaluated their binding affinities for DXS enzyme by SPR. Most of these hits showed substantial improvement in the binding affinities for DXS. And 5 of these hits showed single-digit micromolar affinity. When we evaluated these 11 hits for their inhibitory activities, 6 compounds emerged as a moderate inhibitor for DXS, similar to the hits from the first and second round of DCC, out of which compound number 16 showed the best IC50 value of 34 micromolar. However, 5 hits showed improved antibacterial activity against E. coli, out of which compound number 19 and 20 featured the best MEEK value around 15 micromolar. Overall, the hits from the third round of rationally designed DCLs showed a significant improvement in the binding affinity and antibacterial activity compared to the hits from the first 2 rounds. Moreover, compound 16 from the third round emerged as the most potent inhibitor of DRDXS. To obtain the information about the mode of inhibition of these inhibitors, we selected representative compound 1 for this study, and we perform competition enzyme activity assay where inhibition of DRDXS was measured with varying substrate concentration. So the concentration of cofactor thiamine diphosphate had a strong influence on inhibitory activity of compound 1, so which was shifted to the higher micromolar value when we added more thiamine diphosphate. Pyruvate concentration showed the similar effect, increasing the IC50 value while increasing the pyruvate concentration. Concerning the gap, no influence of substrate concentration on the inhibition could be observed, and all major IC50 values were in the range of 50 micromolar. So from these findings, it suggests that the compound 1 is THDP and pyruvate competitive and non-competitive to the gap substrate. So here we thought that we can also use DCC to confirm the mode of inhibition of the compound 1. So in the literature, it's known that the butyl acetyl phosphonate, which is BAP, binds in the active site covalently to the thiamine diphosphate. And the presence of this thiamine diphosphate or BAP in the DCC will negatively influence the amplification of acylhydrazones if they are a substrate or thiamine diphosphate competitive inhibitors. So to support to this notion, we run two DCC experiments where in one we included thiamine diphosphate and in other thiamine diphosphate and BAP. And we compare the amplification of these 5 hits with the initial amplification where we didn't add any of the substrate. And as you can see, for compound 1, 2, 4, and 5, addition of thiamine diphosphate decreases their amplification to a significant level, which further decreased when we use thiamine diphosphate and BAP. So this study suggests that the most of these identified hits from DCL1 compete with thiamine diphosphate and one of the substrate, and which also supports the mode of inhibition study of compound 1 done by competition assays. So at the end, I hope that I convinced you with our proof of concept study that target-directed DCC can be used effectively as a hit optimization technique, along with its well-established application of hit identification. This process exhibits a potential for identifying potent hits in a less time compared to the traditional METChem approach. Additionally, we also showed that DCC can be used for identifying the mode of inhibition in the competition DCC experiments. So this brings me to the end of my talk, and here I would like to take an opportunity to thank Professor Hirsch and whole DXS team, of course the funding agency, and you all for your kind attention. And I'm happy to answer your questions.